Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your biosecurity, udder disease and bacterial content of raw milk. Now, can you please tell me what is this udder disease? Anybody? Can you please tell me what is this udder disease? You can type your answer in a comment box. Okay, this udder disease are called a mastitis, mastitis, mastitis is common disease which are affect the udder of cow. Is common disease which affect the udder of cow. Okay. Now today in today's lecture we are going to discuss about the biosecurity, udder disease. Okay, and bacterial content of the milk, and this disease are caused by a bacterial infection. Okay, now today we are going to discuss about this. Since humankind began collecting milk from our domesticated lactating animals, which means that we collect the milk from our domestic lactating animal. That means that our milking is done by a human being. Okay, when milking is done by a human being, then there is a higher chances of the contamination, higher chances of the contamination. Okay, now biosecurity and other disease status have been constant animal health concern because we are collecting a milk from our domesticated lactic animal. For many years, researchers have been studied dairy herds and individual cow to gain insight into a causative factor which contribute to a herd health problems. Okay, and their findings reveal three common factors which hurt the health problem. For the first one is your health and lactation age of the animals. Okay, if the health of the animal are poor, then it will also leads to the poor quality of the milk. Okay, and even the lactation stage or are also affect the biosecurity of the milk. Okay, and the second one is your production environment and condition. Suppose that a milkman milking a milk from an animal. Okay, this animal are healthy. Okay, and the milk and the hand of the milkman was also clean, but the environment from but the environment their milkman milking the animal milking the milk are not properly clean. They are infected, like suppose that the bucket in which they store the milk, okay, it is not clean properly, then it will also leads to the biosecurity of the raw milk and the collection practices. Okay, so these three common factors which affect the quality of the milk that researchers find in their research. Okay, now the next one is your with the current trend in United States toward expanding the number of cows in dairy herds, biosecurity has become a major concern as a new cow are introduced into a milking herd. Okay, if we talk about the current situation in a United States, then there is a expanding number of cows in a dairy herds. Okay, when a number of cows are increases in a dairy herds, then the biosecurity are become the major concern because in United States a new cow are introduces into a milking herd. To address this concern, most farm managers are using a coordinated milking unit to increasing incoming cows. Okay, so for this concern, like biosecurity concern, the manager of the most farm, what they do, they using a quarantine milking unit to screen the incoming cows. What they do, they quarantine the cow. They quarantine the new cow for their screening. Issue of antibiotics, vaccination, reproduction, other health, and abomasal disorder are assessed in this unit. Okay, in a quarantine unit, these issues are assessed. The first one, your antibiotics, vaccination, reproduction, other health, and your abomasal disorder before cow are moved into a main milking unit. Suppose that this is your main milking unit, then the new cow will be placed or will be um, stay in a quarantine area where different issues are monitored, like your antibiotics, vaccination, reproduction, other health, and abomasal disorder. Then after that, the cow are transferred from the quarantine area to the main milking area due to the biosecurity concern. Biosecurity concern. Okay, then animals failing to meet the defined health requirement are removed from the herd. While there is an initial expenses during the screening process. Okay, major herd problems can be averted and protocols can be established to assure the expected productivity of the herd. So there are different protocols are assured so that the productivity of the herd can be improved. But even with the healthy cow, dairy producer continue to encounter the challenge of this mastitis in the herd. Okay, even the healthy cow have also challenges for the dairy producer of this mastitis in the herd. Now the next one is your, it has been observed that the bacterial content of the raw milk can be influenced by the presence of mastitis among our dairy product producing animal. Okay, if we observe that the bacterial content which are present in the milk, and this bacterial content are totally influenced by the presence of mastitis among the dairy producing animal. Okay, if the dairy producing animal have a mastitis, then it will increase the bacterial content into our raw milk, and it is said by a or described by a Bramley et al. 1984. Usually, this mastitis or other information is a non-sequence of bacterial infection and is responsible for considerable economic loss. Okay, they do a economic loss due to the lower milk yield. Okay, if a cow have this mastitis or other inflammation, then what they do, they lower the milk yield. Due to this, the economic loss occur. Again, it is described in a Bramley and Dodd 1984. Mastitis may be present in a clinical form in which macroscopic changes to the milk or udder are readily detectable at the time of the milking, and it is described in a figure 2.2. Okay, this mastitis are present in a clinical form. Okay, and it is macroscopic changes to the milk or udder are readily detected at the time of the milking. Okay, when milking are done, then this type of disease cannot be detected, but is more commonly present as a subclinical conditions in which both milk and udder appear are normal. Okay, so subclinical mastitis can be diagnosed only by testing the milk sample. Okay, if we diagnose this mm, mastitis, then it will only diagnose only by testing the milk, which have the result that the presence of the pathogenic bacteria, and this can be described in the next figure, that is 2.2. And it is increase the somatic cell count and a variety of biochemical changes which are sign of udder disease. Okay, if a cow have a udder disease, then it will increase the somatic cells count. Okay, and there is a variety of biochemical changes occur in a cow, and it is described in a we look at all 1962 Scarman et al. 1971. These composition changes largely reflect an increased movement of, now come to the okay. Now this compositional changes largely this compositional changes largely reflect an increased movement of blood component into the milk during a inflammation. The milk from the infected quarter contain increased concentration of the antitrypsin, bovine serum, albumin, immunoglobulin, and sodium chloride ion, whereas the concentration of the lactose and potassium are reduces. Now the next one is your, see, this is the figure which is withdrawal of fore milk from a udder quarter infected with a clinical mastitis, demonstrating severe clotting of the secretion. This diagram describes the withdrawal of the fore milk from the udder quarter infected with a clinical mastitis, demonstrating severe clotting of the secretion. Now, next diagram is the microscopic appearance of the milk from the udder quarter subclinical infected with a Streptococcus aureus, showing the infection of the cocci fibrilin, and it increases our leukocyte count. Now the next one is your, in a many countries employ control system which is used to reduce the incidence of bovine mastitis. The other system can also divide into two types if we talk about in United Kingdom, United States, Australia, and other countries. Control measures are based upon the application of the disinfectant tea dip after milking and the treatment of all udder quarter with the antibiotic at drying off. Oliver et al. 1992. They suggested that the intramammary infusion of cloxazine or cefaferine 7 days before the calving can reduce the mastitis incidence in the early stage of the lactation, and the treatment of these and the treatments with the different antibodies at a drying of 7 days before the calving act both Prophylactically to prevent the new udder disease arising when the cow is not milked, and the therapeutically to eliminate the existing subclinical mastitis when dry and during early lactation. Level of the udder disease have been markedly reduced with this management, which is approached in figure 2.3, and the Streptococcus aguilar city has been eradicated from many herds, and it is said by Wilson and Kingville in 1975. So this is the diagram for reduction in the proportion of cow and other quarter with subclinical mastitis in 30 herds over 3 years using a control system including a post-milking tree dipping and using a antibiotic therapy of case of clinical mastitis, and for all cows at the end of the each lactation. Okay, so this graph are between the percentage of the infect and the start. Okay, this is your 0 year, then starting of the year. This is your first year, second year, and third year. And this is other species. Then this is your staphylococcus or any bacteria. Okay, now the next one is your proper sanitary preparation of the cow teat prior to a milking and post milking teat. Disinfecting help to reduce the transfer of the bacteria during the milking, and it is also destroy the pathogen which is left to the teat in skin on the end of the milking. It is during this time that the teat orifice is open and the flow of milk provide the link between the environment and intramammary gland. Preparing the teeth of the milk include washing the teeth with the combination of the disinfectant detergent and detergent like your iodophor, followed by drying the teeth with the single-use towels. Once the milk has been collected, the post-milking teeth dip into apply to each teeth prior to the cow leaving the milking parlor. Which teeth dip is most effective continue to the debate among the dairy professional. The NMC at its 1994 annual meeting formed a research subcommittee to examine peer review scientific literature concerning teeth disinfectant. It is finding can be found in the internet website, and this is the name of the website. Initially, essentially, 7 disinfectants were evaluated like chlorohexadiene, iodine, linear didosyl benzene, lauryl sulfate, quaternary ammonium compound, sodium hypochlorite, and iodophor. These disinfectants appear to be most effective against Staphylococcus aureus and Streptococcus agalacide. Okay, so these are the list of the disinfectant, and disinfectant are most effective for two types of bacteria, that is your staphylococcus aureus and streptococcus agalacide. Okay, now the next one, an alternative approach for reducing the mastitis in United States, Denmark, Sweden, Switzerland, the Federal Republic of Germany, and other countries employ extensive laboratory facilities to detect heavily infected herds by a bacteriological or cytological examination of the bulk tank milk. Okay, and this approach is very similar to the one employed by a manager of expanding dairy herds. Problem cow are examined in detail, and infected cattle are treated with the antibodies. Okay, suppose that's in a research there is some problem with the cow, then this cow should be examined in detail, and their infection can be treated with the help of the antibodies. Both approaches with consistent management effectively reduce the rate of the streptococcal and staphylococcal udder disease. These approaches, however, are relatively ineffective in preventing E. coli mastitis, a serious problem among a housed cow exposed to a excessive wet condition. Now, this is the table which explains the effect of including milk from a cow infected with A streptococcal and B is for a coliform mastitis on the bacterial count of the bulk herd milk. Okay, so this is a weekly sample taken. Okay, this is a total bacterial count, streptococci, somatic cells. This is for a streptococcal mastitis, and the B for a coliform mastitis. Okay, here are total bacteria, then coliform, then somatic cells. Okay, so this is the data from a cuisine 1978. Pathogens for man in raw milk. According to a Dr. Michael Taylor, who reported to be the first scientist who observed that contaminated raw milk could spread disease in a human being. Okay, he is the first scientist who observed that if the milk is contaminated, then it could spread to a disease in a human being. He also observed through a epidemiological evidence relative to the spread of the typhoid and scarlet fever within a family living in a Penrith, England. In United States, there is a similar epidemiological evidence that raw milk can be vehicle for spreading disease. Armstrong and Paran, 1972. Raw milk may contain microorganisms that are pathogenic to human beings. That means that they will capable for causing the disease, capable for causing the disease into a human being, and their sources may lie either within or outside the udder. Historically, the most serious human disease disseminated by the consumption of the contaminated raw milk are tuberculosis and Virulosis. Okay, there is a two serious disease occur in a human being, that is your tuberculosis and virulosis, which are occur due to the consumption of the contaminated milk. In both disease, the causative agent organism that may be excreted in milk cocci from infected animal are Mycobacterium bovis or M. tuberculosis and Buricella abortus. B. Malatanus, B. Teos. Often with Buricella infection, there is a little change in milk or udder. But in the case of tuberculosis mastitis, a pronounced and a characteristic change in the milk and udder is observed. In many parts of the world, these diseases have been eradicated from the cattle and no longer pose hazard to a human health. Pathogenic bacteria also may be present in raw milk as a direct consequence of the udder disease. Among the organisms commonly producing mastitis, Staphylococcus aureus, Staphylococcus aureus, E. coli are pathogens known to human beings. Streptococcus aureus can initiate a variety of clinical conditions, the most serious of which are a bacteremia and meningitis of newborn. These two diseases are occur in a newborn, which are potentially fatal to the infected fan. So this is all about the pathogens or a disease occur in a human being or due to the consumption of this contaminated food milk. I hope you will understand. If you have any query related to this topic, you can ask me in a comment box, or further detail you can follow the Idealist Academy on different platforms like Instagram, Facebook, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your account, that is 7080833345, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.